So at the outset to begin the seminar, I would request Dr. Shoptoshi Chatterjee, Chief Coordinator and Principal Investigator, Ejuba Center, Adamas University, to lead the guest on the dais. So please. I would also request Dr. And all the deans and all the speakers to come up on the dais. Thank you, sir. So thank you. I welcome you all to the National Seminar on Vigyan Se Vikas, Showcasing Potential Journey and Impact of Biotechnology on Society. Your presence would enlighten the audience and be appropriate to mark the 75th year of independent India, popularly celebrated as Azadi Ki Amrut Mahotsav. So now I would request Dr. Shoptoshi Chatterjee to felicitate our guest of honor, Dr. Kumar Mohandar. So please. Thank you, sir. Next, I would request Dr. Kuntal Pal to facilitate Dr. Shankhadeep Bose, Principal of Pharmacy Program at Sanaka Educational Trust Group. Thank you, sir. Now I would request Dr. Shubhendu Bandyopadhyay to facilitate Dr. Sushil Kumar Mitra. Thank you, sir. Now I would request Dr. Moshumi Shaha, Project Coordinator of Bairak Yuva Center, to felicitate Professor Shondip Banerjee, Dean of School of Agriculture. Thank you, sir. So before proceeding further, I would request Dr. Shoptoshi Chatterjee, Chief Coordinator and Principal Investigator of Ejuba Center, Adamas University, to speak a few words about the center and the entire experience regarding the center. And before that, I would request Moshumi ma'am to felicitate Dr. Orindo Mitro. Thank you, sir. So now Saptoshi sir will be speaking few words regarding the center.
Thank you, Shreya. A very warm good morning to all. Good morning, everyone. So we have been missing this essence in the last 2 years because most of our good morning wishes were concerned because the mic was muted, right? So it is my proud privilege to organize or to be a part of the first physical seminar that you are witnessing in the last 2 years. After a break for a long time, I represent the Ejuba Center at Adamas University, which is one of the 10 such centers in India, and that has been approved and supported by BIRAC, Government of India. We are going to shortly begin the journey of the Ejuba Center in the form of inauguration. So very shortly, we are going to mark the inauguration of the Ejuba Center on 28th of February, that is National Science Day. But today, there is a purpose why we are here. The program is jointly organized by the School of Life Science and Biotechnology, School of Medical Sciences, and School of Smart Agriculture. The reason being, we are celebrating the 75th year of independence, popularly known as Azadi Ka Amrut Mahotsav. Therefore, it is a time for us to look back what all we have achieved and to take an inspiration from that in designing where we want to go in the days ahead. So today we are very much fortunate to have eminent speaker amongst us who would be enlightening you through various biotechnological progress, which is a complete interdisciplinary matter. It's not a classroom of agriculture or microbiology or biotechnology. It's an integrated approach that is created to serve the society. So with this note, and wishing you a very enlightening event for the day, I would also thank the speakers for their gracious presence, and I hope this program will enlighten you and help you in the days ahead. Thank you very much.
Thank you, sir, for motivating us and encouraging us. And we are looking forward for today's session. So before that, I would request all our dignitaries to please take the seat so that we can start the lecture sessions. So now I will start the lecture sessions by our guest. So the first session will be taken by our guest of honor, Dr. Kumar Mohandar. So for more than 3 decades, he has been working on the molecular biology of Leishmania donovani, the causative agent for visceral leishmaniasis. He has made significant contribution in biochemistry of DNA topoisomerases of Leishmania with reference to development of therapeutics. His area of research includes basic DNA enzymology of topoisomerases, development of anti-leishmanial compounds targeted to DNA topoisomerase, cell death of the parasites induced by DNA topoisomerases inhibitors, and development of kinetoplast DNA-based PCR diagnostics. Apart from this, he has received numerous honors and awards. To name a few of them are Raja Ramana Fellowship by Department of Atomic Energy, Government of India, Nitya Anand Endowment Lecture Award for Outstanding Work in Biomedical Research, including New Drug Development by Indian National Science Academy, Sir J.C. Bose National Fellowship by Department of Science and Technology, Government of India, Deshratna Dr. Rajendra Prasad Memorial Lecture Award by Rajendra Memorial Research Center Institute, ICMR, Patna. So now I would request Dr. Hemanta Kumar Mohandar sir to please come and continue the lecture session.
স্লাইড দুটো স্লাইড এখানে রাখা আছে স্যার একটু দেখি তো দুটো স্লাইড
ঠিক আছে স্লাইডটা কোন লাগবে না জল আছে
জল আছে
ভিডিওটা অফ আছে
এটা অফ রেখেছি তো আপনার তো ফেসেস ছিল ওকে কোনটা সেটটা আসলে শেয়ার অন থাকলে হবে না শেয়ার অফ করে
শেয়ার অফ
এখান থেকে স্ট্রাকচার ভিডিও অন
Now they taken the second set of slide.
sir এটা
No, এটা না
তো এখানে মানে কি করে ওখানে তো মিলটা করে আর কিছু তো আমি আর একটা ছিল
sir he said I will call you once this is
connect করতে
This is the first page.
sorry sir
হ্যাঁ ঠিক আছে এই এটা এগুলো দিয়ে কপি না একটা না দুটো আরেকটা পেজ নিমিশা দুই
Second set.
আমার পাঠাচ্ছি দেখো কোন কোন
আপনি দিলেন স্যার
দুটোই হ্যাঁ দুটো ট্রেডমিলে দুটো ছিল এটা দেখো
11:21
দেখি না এটা দিয়ে শুনুন ওইটাই ওটাই আগেরটা ওটা ছিল না এবার এই দুটো আলাদা নেন আপনি কি
এটা তো এলো না।
ওখান থেকে খেলাম আমার থেকে খেলাম কি করছো
এটা তো এটা
এইটা ডাউনলোড
ফেল্ড।
এটা তো এইটা
হ্যাঁ স্যার, ডাউনলোড ফেল্ড হচ্ছে।
হ্যাঁ 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 হুম
স্যার দুটো পরপর কানেক্ট করতে হবে।
হয়ে গেলে
জিজ্ঞেস করো
এইটা
সেইটা
এইটা তো বারবার
এইটা না
আসছে।
হ্যাঁ তাহলে আগেরটা দেখো এখানে আরেকটা আছে দাঁড়াও, দুটো সাইডের একটা সাইড বেরোচ্ছে আচ্ছা
প্রথম সেট অফ ফ্লাইটে গেল
আচ্ছা না না ঠিক আছে দুটো এখানে
স্ক্রিনটা শেয়ার।
খুলে
স্যার মেলটা
দুটোই আছে দুটোই পাঠানো কোথায় খোলা নেই এখানে
এইটা না স্যার।
সবার করে দিয়েছি
আসেনি তো।
এই তো তুমি এটা মতো কি আসছে এই তুমি এটা মতো দুটোই তো আছে এটা মতো হ্যাঁ দুটোই আছে দেখো হ্যাঁ এটা এটা অন্য সাইডে এইটা
এটা ফার্স্ট তারপর ওইটা
হ্যাঁ ঠিক আছে পরেরটা শেয়ার করো হ্যাঁ দুটো পেয়েছো না
আচ্ছা এখানে ও আর এটা নেই তোমার কোনো পয়েন্টার
পয়েন্টের জন্য এটা দিয়ে স্লাইড চেঞ্জ করতে হবে।
ফোকাস যদি করি কিছু লাইট লাঠি ফাইট
স্যার ফোকাসে কিছু আছে পয়েন্টার। Sir, I can. No, sir wants to focus something on something.
সিম্পল একটা জায়গায় ছিল লাঠি হলে এনিওয়ে সো কোন দিকে এটা হ্যাঁ
এটা বাঁ দিকে টানছে স্যার স্লাইড
বাঁ দিকেরটা হচ্ছে
হ্যাঁ ডান দিকেটা করুন স্যার ডান দিকে ডান দিকটা করলে এটা কি স্যার পিডিএফ এ আছে
PowerPoint.
ঠিক আছে এই আমার থেকেও তোরা খারাপ
হ্যাঁ হ্যাঁ ওটা হ্যাঁ
ছেড়ে দিন আজ নেক্সট স্লাইড করে আমি চেঞ্জ করবো এই তো ওকে ফাইন should I start মাইকটা ঠিক করে দাও। Hello, can you hear me? Good morning, everybody. It's a real privilege for me, and I have been honored for this invitation from Adams University. I've been invited to speak about the biotechnology, the subject. People think that it's a new one, which I do not believe. The biotechnology is there. It was there. It is an ancient subject. Everybody knows that during the civilization, long ago, people used alcohol fermentation to make drinks. People used to take cheese, curd, by fermenting milk, and so many products, food products are there. People produce hybrid, cultivators produce hybrid plants and hybrid animals also. So it's not a new subject, it's an old one. Biotechnology is a subject. People consider it in a different way. See, I will tell you briefly, I'll make a brief history, principles of, and everybody thinks that biochemistry, biotechnology is nothing but recombinant DNA technology. You know, recombinant DNA technology is one of the part of biotechnology. Like alcohol fermentation is not a recombinant DNA technology, but it is biotechnology. Molecular tools of recombinant DNA technology. While speaking on this subject, I will be concentrating on the recombinant DNA technology, a part of the biotechnology. I remember when I was doing my postdoc in the United States in 1976 to 80, and then I moved. At that time, recombinant DNA technology was not born. And then I moved from Albert Einstein College of Medicine, New York, and I went to University of Zurich, Switzerland. I learned biotechnology, recombinant DNA technology there. And that was in 1980. It's long back, 41 years ago. I cloned the gene over there, expressed the gene in large scale, and then purified those products and used in the market. What are the main ingredients of the recombinant DNA technology? Is restriction in the nucleus, and then preparation of DNA, sequencing of the DNA. I remember the DNA sequencing has come to it, and then next generation sequencing, and then other sequencing processes, then came PCR, all simultaneously after the advent of this recombinant DNA technology initial experiments. So while talking about the recombinant DNA, I am telling you just the principle, then I will come to some of my experiments, which is really the product of this recombinant DNA technology. What are the host cells? Host cells are, I'm a person, I have been infected all the time. Everybody is infected. So infection comes to the host. We are hosts. What is vector? Vector is the carrier which takes the material, the infecting DNA into our cells. Bacteria is a host cell, yeast is a host cells, and then protozoa is a host cell. But the vectors are plasmids is one of the vectors, Viruses are also the vectors. So recombinant DNA technology consists of many experimental things which are being advanced by the presence of these vectors. So what is transformation? Transformation means change. Like suppose you have a bacteria, you are growing the bacteria, and then this bacteria will grow as long as it gets the food. And then if you add some antibiotic, the bacteria will die. It will die because it cannot resist the killing property of this antibiotic. But some bacteria naturally they acquire some small DNA molecules which are called plasmids. These plasmids, when present in the bacteria, they can be removed from the bacteria also. These plasmids, when present in the bacteria, they induce some of the properties like ampicillin resistance. Some vector plasmid contains the gene for this ampicillin resistance, and so if you add antibiotic into the bacterial culture, it will die. And then if you introduce the vector system or the plasmid into the bacteria, coding for this recombinant plasmid, coding for that resistance gene, it will survive. That is the way selection through bacteria and antibiotic goes on this way. So what are the applications of recombinant DNA technology? I will tell some of the facts. Recombinant DNA technology application means, say for example, you want to grow some compound in a large scale. You cannot do that. But if you take the help of recombinant DNA technology, you can produce large amount of that product you need for. So what happens if you want to grow, for an example, if you want to have insulin can only be available through animal slaughtering. From the pancreas of the animal, you can isolate the insulin. So when discovery of diabetes has taken place, so that was a time the pancreas of the animal bovines, they have been taken out, they homogenized, took the extract and used that extract for treatment of the diabetic patient. But now you cannot do that because it will come down to the level. Somebody speaking, please. Sorry, that disturbs your crosstalk. Anyway. I am the hybrid DNA molecule created by cutting and shelling. When introduced into the host cell, I multiply and code for the desired proteins. So vectors, with the use of recombinant DNA enzymes, technology enzymes, restriction enzymes, ligase, DNA, everything. So mix together and get it ligated, and then you put it into another bacteria, which will be transformed. So his nature will be changed. And that nature, you are going to utilize that nature for development of this protein. The present day DNA technology has its role in the experiment performed by Herbert Boyer and Stanley Cohen. I have seen both of them. Before Boyer and Cohen biotechnology. Before Boyer and Cohen, they are the pioneer. They discovered this recombinant DNA technology introduced, but after that, so many other people are there who are very much involved in that. And after Boyer and Cohen biotechnology, what was the status? That is explained in the book. So now, this is the principle of this recombinant DNA technology. See what happens. This is the plasmid vector, which is, I already told you, is present in the bacteria itself. and it is an extra chromosomal element which is self-replicating. Okay, you can isolate the plasmid from the bacteria, naturally occurring bacteria. So it's a circular DNA molecule, and on the right side is a DNA fragment. Okay, you want to join this DNA fragment out from outside into the plasmid vector. I think this will be better. Okay, so to cut this, the principle of recombinant DNA technology, plasmid vector plus a DNA fragment, you mix them together, nothing happens. Now, if you take that plasmid DNA vector and cut with a restriction enzyme will cleave that DNA, and then you join that DNA fragment, green color, by an enzyme which is DNA ligase. So you are ligating that green colored DNA into the vector. So now vector cannot distinguish its own DNA from the foreign DNA. It will use it as its own. So take this recombinant plasmid. Now, if you take this recombinant plasmid and put it into bacteria, this process is called bacterial transformation. Simply, you take the bacteria, grow them, and in presence of calcium chloride, the membranes of the bacteria will be disrupted, and then you add the foreign plasmid vector containing the recombinant DNA, and then you grow it. So suppose look at that. This is the left side is called recombination has taken place inside the bacteria, and on the right side there is only the bacterial chromosomal DNA. Next, introduction of recombinant DNA vector into host cell. Then you put it into the host cell and plate it, and you will get those principles. Sorry, I am not habituated to this type of juggling. Okay. I did my PhD in 1975. Many of you are not born at that time. But anyway, I have learned something. But this jugglery again disturbs the lecture. Molecular tools of recombinant DNA technology. Anyway, what I'm telling you that restriction in new nucleus, the name you know why it has come. It is E. coli. E. coli produces some enzyme for its own survival. If a virus infects any bacteria, E. coli, then what will happen? E. coli will definitely try to kill the foreign DNA, foreign viral DNA. And for that, how to kill? E. coli produces an enzyme which is for restriction enzyme, EcoR1. So all the bacteria, they have their own machinery to produce enzymes for their own survival. For example, BAMH1, have you heard of BAMH1? The BAMH1 Bacillus amyloliquefaciens, the host, from there. Then HEI3, Haemophilus aegypticus, the bacteria. You isolate the enzyme HEI3. SAL1, Salmonella albus or something like that. So many restriction enzymes are produced from so many bacteria. And those restriction enzymes have got the characteristics. It's not my fault. I'm prepared to give a good lecture. So it's not my fault because your technology I don't trust technology. Your technology disturbs always, always. Whenever you ask when I ask my daughter, can you add 25 plus 37? She will take a computer and do it. I tell them that, look, I can do by myself from my memory. So that technology is so advanced and we are so poor. Anyway, I have some good slides, very interesting slides. I want to show those slides. I have been working after my postdoctoral studies. I joined Inauguration Clinical Biology in 1982. Okay, so almost 40 years. And I was asked, I learned the recombinant DNA technology. I could do cloning over there. But I asked, our director told me, you have to develop the molecular biology of the Leishmania parasite. You have heard of the parasite, it's a protozoan parasite, Leishmania. It causes sclerosing. Similarly, malaria is Plasmodium, Plasmodium pilex. They cause malaria. African trypanosome is a protozoa. It's very dangerous disease. These are called parasitic protozoa. So I developed an institution here within IIC for research on Leishmania. So I did. And I have done some work which I'm going to tell you right now. Invite me a model lecture to me.
So not this one.
Not this one. I could do parasites own flavor.
Okay, another presentation.
He is senior to me by 10 years. Same department, 10 years, but we are very, very close. He just passed away.
Screen share আসছে না
ওটাই তো বলছি যে স্ক্রিন করবেন যেটা চলে ওইটা স্ক্রিনশট দিন কোনটা
এইটা ওখানে তো আসছে না
ये तो ছিল না প্যারাসাইট সমস্ত
হ্যাঁ স্যার এখানে খুলছে কিন্তু এখানে শো করছে না সামহাউ স্যার এই স্লাইডটাই তো বলছেন যেটা একদম স্টার্টিং এ খুলেছি this one
কি লেখা আছে
এইটা বাট এখানে সামহাউ আসছে না
তাহলে কি হবে
মানে আপনি জুমটা বন্ধ করে এমনি স্লাইড শেয়ার করুন
খুব ভালো ভালো ছবি আছে
এই স্ক্রিনে আসার জন্য কি করতে হবে সেটা করুন লাইভটা থাক স্যার এখানে স্ক্রিন শেয়ারের ব্যাপারটা তো আমার দেখান না
লাইভ হচ্ছে এখানে সেকেন্ড স্লাইডটা বের করো যেটা সবচেয়ে পরে একটু এখানে এই তো কথা তো লাইভটা কাটো
লিভ মিটিং হ্যাং হয়ে যাচ্ছে এই স্লাইডটা
হ্যাঁ এই স্লাইডটা
good purpose when there is a oil slippage in the ocean. So just spread that bacteria. It will use up all the oils and they will so I have also produced
to extend that
recombinant DNA has already come 1975.
আর ওই এটাও তো ফেরা এবার স্লাইড শেয়ার করুন শেয়ার করতে হবে
Gilbert. So many.
ওইটা তো যোগ করে
You just change the slides when I will tell you, and then I have to cut down my slides because this is introductory slides. I didn't know. it would have been nice if I could show them, but anyway, time is very short.
এটা চেঞ্জ হচ্ছে এবার
হচ্ছে এবার
এই যে এই এটা দিয়ে
ঠিক আছে আমি
ঠিক আছে।
আর দরকার নেই, আমি এটা দিই The title of the slide is Parasites Are Smarter Than Humans. This is the work being carried out in my laboratory along with others. Because so it is from infectious diseases, the immunology division of CSR, which I was heading the division for last 15 years before my retirement. Issues to worry. Malaria caused by the parasite of Plasmodium genus tops the list of the global death burden occurring due to protozoan parasite. WHO has reported 228 million cases of malaria with 405,000 deaths in the latest malaria report in 2019.